In this lecture, we're going to talk about microscopes. A simple microscope is just one lens that you use to enlarge an image. So sometimes that's often also called a magnifying glass because there's only one lens that you look through to make things look larger. A compound-like microscope is the kind that we would typically use in class. This is when you have a series of lenses, typically at least two, and light travels through the object and then through the lenses to enlarge the image. When you use a compound light microscope, the subject can be alive while you're looking at it, which makes it different from some other types of microscopes. So when you look at the diagram here, you can see at the bottom of the microscope is a light source, and that shines up through this stage, and typically you put the sample that you're looking at on a glass slide on the stage right there. And then you have some objective lenses. Usually they come in multiple magnifications, so this one has 3. And those 3 lenses allow you to view things larger when you combine them with the lenses that are in the eyepiece. So that's why this is a compound light microscope. The light travels from the light source through the subject, through the first lens, and then through the objective, or through the eyepiece lens. And so those 2 different lenses work together to allow you to view your subject more easily. When you look through a light microscope, a lot of the images will look kind of like this. This is an organism called a paramecium. And paramecium are single-celled organisms that live in pond water. So if you were to look at water from a pond or a lake under a microscope, you'd see probably some of these guys swimming around. You might notice they kind of have this fuzzy edge. That's because they have structures called cilia that help them to swim around and to move so that they can collect food and things like that. Here's another image of different organisms that live in fresh water that you can see through a light microscope. So these are different types of algae, which means they're photosynthetic. That's why you'll notice they're green. So they use light from the sun, and then they collect nutrients from the water, and they can use that to make food, and they can live in water of various temperatures. They can even survive the winter in like Minnesota lakes if they don't freeze in the ice. There are also electron microscopes. They don't use light. They, rather than light, use a beam of electrons, and it goes through or around the object you're looking at, and then the image is produced on a fluorescent screen. When you use an electron microscope, the specimen has to be in a vacuum. When something's inside of a vacuum, it means that it's inside of a space that does not contain any air. They suck all the air out. Because of that, the specimen you look at has to be dead. So you can only look at dead cells under an electron microscope. That's different from a light microscope, remember, where you can look at live specimen. There are 2 different types of electron microscopes. So one of those types is the scanning electron microscope. Sometimes those are also called the SEM. And they send a beam of electrons around an object. And so you can see the surface detail of an object super easily. So this, for example, is the cross section of a leaf from a walnut tree. So this is a leaf. This is what a leaf looks like when you look at it through a scanning electron microscope. And you can see in great detail the vein of the leaf that is here, and you can see it's made of cells, and then you can see some of the cells of like the palisades layer. All these little tall cells are cells that from one of the leaf layers. This is a scanning electron microscope image of pollen grains. So if any of you have allergies, if you've ever had pollen from trees or from grass in the spring or the fall make you sneeze a lot, this might be what you're allergic to. Pollen is the part of the plant that carries sperm cells from the male part of the plant to female flowers, and that's how plants reproduce. And then this is a scanning electron microscope image of an American cockroach. You can see this is his head. So these are the antennae that stick out from his head, and then these are the mouth parts here. And so that's what a cockroach looks like when you get up really, really close. The other type of electron microscope is the transmission electron microscope, or sometimes those are called a TEM. and they shoot a beam of electrons through an object. So they allow you to see great detail on the inside of a cell or for something like that. So in this image, this is a picture of the Golgi apparatus, one of the organelles in a plant, and it comes from a corn root. So within one cell in the corn, you can see this kind of lumpy, wiggly structure here. That's the Golgi apparatus. In this transmission electron microscope image, this is where you can see what a bacterial cell looks like. They are a lot simpler than eukaryotic cells because they're prokaryotes. They don't have a nucleus. They also don't have membrane-bound organelles, and so they're a lot smaller and they have a lot less structures on the interior of the cell. Inside of your cells, you have an organelle called the mitochondria. So this is an image of a mitochondria. So here you can see it kind of looks like a bean. And that is the mitochondria, the little powerhouse of the cell that's found in both plant cells and animal cells.